Thank you, Michelle. That was a very kind introduction. I'll try to live up to it. Right, so like Michelle said, I'm Blake Sweeney. I kind of handle the day-to-day stuff for RNA Central and RFAM at the EBI. And I'm here to talk about some of our vision and then maybe a bit of what the broader field's vision for the future of RNA tools is. And then afterwards, we have a panel discussion where I hope to get some very interesting questions and insight from the panelists. So for those of you who don't know, this is our very standard slide for RNA Central. What is it? It's a hub of non-coding information about non-coding. It's a hub of information about non-coding RNA sequences. Effectively, it's a database of databases where we go to a lot of different specialized databases and we integrate them all into one unified view. We now are up to about 45 million sequences. And if you're kind of asking, what do people use RNA Central for? It's like a source of truth. What non-coding RNAs are known? Well, you could go and look at 50 some odd 60 databases, or you could just download it from us and be done with it, right? That's kind of the use of RNA Central. And so if you're asking, what's the goal? What's the vision? Why RNA Central? Well, where are we going? So we start out by getting sequences and metadata, which is kind of just what I described. We then analyze them. So people will tell you things like, this is a human long non-coding RNA. And we align it to the genome to find out, if it actually exists in that, because that's not always true, as it turns out. We analyze sequences with RFAM to kind of give some functional information. And then we start integrating things I think of as more biological or maybe richer. So interactions, gene ontology annotations I find very valuable. I'm going to talk about later. Orthology, where it's available, though it often isn't. And then, of course, this is RNA. So what would RNA be without some sort of secondary structure adding? And so we built a tool called R2DT a few years ago, which visualizes secondary structures in a standardized way. We're very proud of that. And then more recently, we've started looking at literature and publications because our end goal is gene function. We want users to go to RNA Central, find whatever relevant RNAs they care about, however they do that, sequence search, text search, whatever. And then they should be able to know, what does this do? What is this? What does it do? Because the long-term vision, the long-term goal is to be uniprot for RNA. We're not there yet, but we do, we think we've been working towards it. But there's some major, there's a big, huge gap between protein and RNA. Not just the amount of resources and money in it, but it's like missing data. So this is a uniprot page. I literally clicked human and went to the first entry. I have no idea what this is. But you can see here, this box, this text box of function. What that is some number of curators sat down, read a lot of papers, and summarized what's known about this protein. That's incredibly valuable for people and kind of is ultimately what RNA Central wants to provide our users. But there are no curators in RNA Central. It's very unlikely there will ever be any curators. In general, RNA has very little curation activity in terms of people who are committed and focused on just it. It tends to be done sort of incidentally in model organism databases. So the likelihood of us ever having comprehensive literature summaries or summaries of function is from curators is basically zero. It's not going to happen. So what Andrew Green did is he built this tool LitSum to summarize literature for RNAs. It was published a while ago. Basically look for literature where there's a mention of a specific RNA. RNA Central has a very comprehensive set of aliases for RNAs. And so we look across all open access literature. We then do kind of a topic modeling approach, which is maybe a little unusual nowadays. So LitSum is a little outdated, where effectively we look at all sentences which mention an RNA and we pick representatives as needed. And we summarize it with ChatGPT. We do some automated checking with ChatGPT. Basically, you can tell itself, you wrote this sentence and it makes this claim. Does this text support that claim? And it will correct itself. It's not perfect, but it does catch its own errors to some amount. And things like that until we produce finished summaries, which you can go see on RNA Central and RFAM. And so, this is kind of a little retrospective. If we're talking about the future, though, LitSum is technologically very outdated. It was built over a year ago, which means in the LLM world, it's useless, right? So we don't want to do this sort of very fixed pipeline with sentence selection and topic modeling. We're expecting to do some agentic-based summarization Likely internally at least, I wouldn't be surprised to see things by the end of the year, and it'll take longer to become public, but that's absolutely the direction that the resource is going. And there's more, curation activity that's not being covered. So I don't know if you know about gene ontology annotations, but basically they're a structured, machine-readable way of making statements about what a function of something is. So I went to that UniProt page from before, and I just scrolled down a little. I actually had to crop it out of the original picture. And this is some curators have read as much literature as they possibly can about that protein and have decided kind of what its function is. Again, there are no curators in RNA, so we need a solution, right? So we can't do what's normally done. Curator reads a lot of literature and produces annotations. I don't know if anyone saw Chris Mungel's talk, but curators do a lot more than this. I'm sort of simplifying it down to its very, very simplest thing they do. And again, this is work by Andrew Green, where basically we're replacing the human with an LLM, and it does work. I'll kind of summarize that right now. So for microRNAs, the curators have thought very, very carefully about what does it mean to annotate function of a microRNA? What experimental evidence do I need? What combination of evidence do I need? What decisions do I need to make? All that sort of stuff. And if you go to their wiki, which is publicly available, and you scroll down the great deal of documentation they have written, there's this flowchart. And this flowchart is the recent official way of how you can take a paper that you believe informs you of what the function of a microRNA is, or specifically what genes it may or may not regulate, and make that decision correctly. All you have to do is go down and answer a series of yes or no questions. So you read the paper and you go, does it have experimental evidence? Well, no, I don't think anyone can. Let's imagine a paper doesn't. So it goes over this direction and it says it's a computational prediction only. Yes, don't use it. You can't annotate it. Curators demand, quite rightfully, clear experimental evidence. So let's imagine it did. Well, then you can ask, is there an assay for the effect on the target microRNA expression? And depending on if it's a yes or no, that will change the type of annotations that are possible. Curators have documented which assays are acceptable, which are not, exactly what needs to be done. So this is great training. So if you're a new curator, right, you can read this, you can read the documentation, you can understand what to do and get up to speed on a complex task really quickly. On the other hand, if you have an LLM hat on, that's just a bunch of prompts. That's all that is prompting strategy. And so what Andrew did, again, his work, used a local model QWQ32B and effectively encoded that entire flowchart into a series of yes or no prompts. There's some really cool things that this does that I want to point out. So one thing that I think is really valuable is we use constrained decoding, which means you can guarantee that the LLM will only produce tokens you want. So more specifically, you can ask it a yes or no question and guarantee it will only produce the word yes or no, which eliminates, say, some post-processing steps, which is fine. On the other hand, you can also say, find me the part of the paper which supports this claim you made, and you can use constrained decoding to guarantee that it will never hallucinate and will always produce a chunk of the original paper. This turns out to be very valuable and very useful for curators, as I'll show later. I'm not going to go through all of the sort of initial validation stuff. I'm going to talk about some of the more exciting validation at the end. But the end result is with about 3.5 days of compute for GoFlow, you can process about 8,000 papers, get 2,600 Go annotations for about 1,000 microRNAs. Each microRNA may regulate more than one protein. And at current rates of microRNA annotations, that's 20 years of effort. So I think techniques like this are basically the only way microRNA or non-coding RNA science is going to catch up to protein. If you want enough data, we're going to have to automate it. And as I'll show, this has acceptable error rates in the eyes of curators who are quite strict. So what's the output? Before we get into anything detailed, it produces these text summaries, which you can read. They're reasoning traces in LLM language. And so you ask it your question at the top, and then it goes through and does its reasoning, gives some its evidence. It will even pick out the exact sentence that you asked for. So this is really valuable for lots of reasons. One, no human is ever going to produce this. Curators will never have enough time to write an essay for a small essay for each time they make a decision. It's impossible. And so this means that when you have them and you're a human, do you trust LLM output without reading it? You probably shouldn't, frankly. But when you need to validate these things, you can trace through this and it gives you its exact reasoning. It tells you the sentence. So it provides very strong support. It makes it a lot easier to validate. So let me show you one particular example at the end. So it was asked, basically, does this have an assay for this SNES3 microRNA expression? It decided no. And the reason is, the evidence is this sentence SESN3 levels were decreased by a mere 780 in both NEP cells and control NEP cells, figure C. So that is a sentence from the paper, guaranteed. It will always be true, which is really useful because curators can very quickly evaluate this. It doesn't have to be a sentence. It could have been a paragraph. It could be a snippet. It kind of depends on what the LLM thinks is the best. But like I've been alluding to, the most important thing is evaluation in ML work. That's like the main thing, is how do you know it's right? And very tragically, as far as I'm aware, the only way to evaluate LLM work is to read it, is to have informed, intelligent human beings sit down and read the answers, which does raise some questions about the long-term utility of them, but maybe we can discuss that later. So that's what we did with this work. We actually co-developed Litsum with 4 curators: Helen Ettrill, Valerie Wood, Julia Antonazzo, and Simona Panini. Maybe some of you know them. And basically they went and read a sample of our, they did 2 things. One, their set of papers, which are already annotated, right? Can we reproduce them? They read that sample. We do very well. I'll discuss it at the end. But also we took papers which have never been curated. No curator has ever looked at them. They're from all sorts of crazy organisms that don't get any curation attention. Viruses, all sorts of stuff. We ran GoFlow on it. And then we asked curators, does it work? Do you believe its decisions? And so here's a little summary of them. So there's 3 terms that it can be assigned to. The terms are in a hierarchy. Here's the blue one is the most generic term. There were 8 annotations GoFlow made in the sample. Curators agreed with all 8. And I don't mean that this isn't like a light test. This is actually quite difficult. They agreed with every decision that the LLM made. So the overall accuracy of traveling the entire flowchart for generic terms is 100% in this sample, which is quite difficult. In this term, which is microRNA-mediated gene silencing by inhibition of translation, there are 10 annotations, and they agreed with 5 of them. The main problem we've seen is it gets really confused with qPCR results. It seems to be both maybe qPCR is confusing to the LLM, but also, according to Andrew, those sections are always really badly written. I don't know if you remember Amos's talk about why these people put all this valuable data in these really awful papers that you can't make sense of. It turns out the LLMs will also struggle to make sense of them. So that's tough. That's tough. And then this final one, which is actually the most common term. It made 42 annotations, and of them 39 were correct. Again, qPCR and confusion across multiple experiments. So in total, this is about 87% accurate, which is, I think, fairly good for this sort of work. For reference, if you say, well, there already is existing Go annotations, does this reproduce them? And that is, it does, but there's a little caveat to it. I'll point out at the end in a minute. So the future, what's the future? I've kind of drawn the person separate from this curation process. That's a bit hopeful. So I think there's going to be a mix of a human in the loop in a sort of co-pilot sense. Maybe you run something like GoFlow and it spits out all the text, and then you click, yes, I agree, no, I don't agree. And then it kind of is considered curated because a human has looked at it. On the other hand, it could be that it's fully automated and just dumped in. There was actually a period during GoFlow's development where we did discuss that with the Go consortium. but we did settle on sort of not having it marked as a normal human curated thing yet. How separate the human will be from this reading the paper step, I think is up in the air. It's going to depend on the domain and the engineering resources dedicated to it. But I think the days of people reading papers very carefully for these sort of annotations is going to be coming to an end. GoFlow has a lot of limitations, which I'm happy to talk about in the question section. But it absolutely captures the common activity at an acceptable rate. And there's one advantage for using something like GoFlow. So this is a table of accuracy over time. So papers that have been annotated with gene ontology terms, they go back to 2015. And what's GoFlow's accuracy on them? And you may be a little concerned that perhaps I've been lying to you this time about its accuracy, because you can see numbers like 33% in 2015, which is not good. It's very bad. And it's only until like 2022, 84%, 86%, that the accuracy rises to something you'd consider tolerable. It turns out that curation standards change over time. So curators, as they better understand the field, as they refactor their ontology, as was discussed in the ontology section, the standards of how you do the annotation change. And this is actually where GoFlow shines, we think, because you can redo the flowchart and then just give it to the LLM and have it redo the entire analysis for you. So you can actually keep your annotations up to date. Another way of looking at this chart is not that GoFlow is wrong, is that all these annotations in the past are not the same in a fundamental sense as the new ones, and the annotations themselves are incorrect. Because when we went back through and we had curators check things from the past, specifically from the past versions of annotations, GoFlow produced the correct modern one again about 90% of the time. So the term we've been using is deepening, because generally those annotations were very generic, but nowadays there's a more precise term that's expected to be used. So I think things like this are kind of where this tool will really shine. And frankly, I think that's kind of the future of Go annotations. It's going to be heavily driven by LLM work. That said, I might be a bit more hopeful on this than others. There are some shortcomings that GoFlow can't do. For example, these papers often have more than the set of annotations that I discussed, the 3 Go terms. And GoFlow can't capture that yet because there's no flow chart for them. But I think that's solvable. We shall see. And so then RFAM is the other resource. And just a general introduction, RFAM is a collection of families. Each family is a multiple sequence alignment, which has a consensus secondary structure. And that's used to produce a covariance model with Infernal, which can be used to find new examples in the genome. What's it used for? Annotation in genomes. Basically, if you're looking small non-coding RNAs in a genome, it was RFAM. It's a reference set as well, and also a training set for algorithm development. So AlphaFold used RFAM as a source of truth. It's very common to do. Kind of some recent work is we're integrating 3D structures. So we're making RFAM richer. So this is an old FMM riboswitch, where after adding a 3D structure to the alignment, we were able to find new base pairs that were only observed in 3D and not the alignment to improve it overall. This is something that Nancy has been working on for years. And Nancy Ontiveros is the curator for RFAM. And so where coverage is around 50%, I actually think a lot of these other ones are maybe not so curatable. And not so interesting. I think we have all the major ones, all the really important ones. So what's the kind of the future for RFAM? RFAM needs to grow. It's very small, as I'll discuss. And basically, we're thinking through clustering in RNA Central. We can build an RFAM-B, which is automatically built families, open that up to the community to add things to as well with very limited curation. So that'll grow it. It'll be of questionable quality because it's automatic, but then it'll still be curated into the proper RFAM. So it'll maintain its high standards while we do that. If you want to know more about this stuff, Andrew had a poster. It may still be up. You can find him and talk to him. Now I want to talk a little bit more about the future tools. So my background is RNA. My PhD was RNA 3D structure. I've moved into sequences, so maybe it's not too surprising. I really wish there was an AlphaFold for RNA. I really wish it worked. I would love to have a structure for every single sequence in RNA Central, where that is a reasonable statement to make. I and some others have written a review as to kind of why won't work with the current set. You can read the paper if you want. But I think it can summarize it very quickly. So in RFAM, there's about 4,178 families. In PFAM, which is a roughly equivalent research for protein, there are 24,000 roughly at the same time. AlphaFold was trained on the BFD, which has 60 million. So 4,000 to 60 million is a considerable gap. Now, this time though, I'm lying to you a little bit. Slightly unfair comparison, okay? RFAM is mostly whole RNAs. PFAM is mostly actually domains. So you can imagine you can have 2 domains and just swap them and get a new protein, right? So it's a bit more combinatoric to get to 60 million from 24,000, but still the gap is quite substantial. If you start looking at the data that's in RFAM, so this is what type of families there are. The most common is a microRNA. It's about 35% of our data. Spiritually speaking, that's like an alpha helix in protein, because they're just very simple helical pairs with a few mismatches. I'm not sure that tells you much about structure. That doesn't tell you anything about junctions, right? Junction orientation is essential to your overall topology. So it's a bit tough to learn a lot when a chunk of your data is kind of basic. And they all look kind of the same from some perspective. If you look at what's in the sequences, what sequences we have, the distribution follows a very different pattern. So the most common are cis-regulatory elements in bacteria. So there ends up being a very practical problem of how do you sample across the space to get a separate test and train set? It's, it's not unsolvable, to be clear. I don't think any of that is unsolvable. I think it's just, time. It's a bit of time, a bit of people who understand this data should work with the structure prediction ones to try to build something. Yeah, I expect it to be solved. I would like it to exist. I think it will during my career, but when? Okay, who knows? Yes, so I think it's much broader than just kind of the view I have. You could talk. There's a lot of companies. So these are just the people I follow on LinkedIn, basically, and they're all excited about RNA. They're doing lots of different things, and we're going to see some great talks about that. So we'll start off with a talk on RNA modifications, which I haven't really talked about at all, but is like totally essential, right, to RNA function and structure. That has to be part of the future. So we'll hear a nice talk about SIMODEM. RNA design, as we've already heard, right, that's with this therapeutics, it's definitely going to be part of therapeutic creation. So we're going to get 2 talks on that, second and fourth. And then microRNAs, despite an extensive amount of study and recent prizes for them, we don't quite understand exactly all the rules of how they work. And I like this talk because explainable ML is something that I would like to be part of the future as well. So that's the final talk. And then we have a panel discussion to discuss these ideas and more. I haven't talked about structure probing at all. And surely that's part of the future, right? And then, of course, the need for benching and various limitations in machine learning. Yeah, thank you. Let's enjoy that. And some self-promotion now. We're going to be hiring bioinformaticians soon. I thought the job ad would be out now, but it hasn't quite been posted. So if you're interested, please keep your eye out or come talk to me. And then thank all of you and thank our funders and the organizers for allowing me.
Hi, so one question I had, so I also started working on RNA 3D structure. I moved from the protein field. And one thing that I was warned is that RNA a lot of times have very flexible 3D structures. So I wonder, and then I also realized that there's a lack of definition for ground truths. So I wonder if you could elaborate on that a bit. Thank you.
So solving the structure, yeah, is quite challenging because it's very flexible. So the things that have 3D structures kind of hand-wavy look the same. They're globular. You don't tend to see a lot of weird extended things. And then what was your second one? Oh, the ground truth for RNA structure. So, okay, this is my bias, and I would happily admit it. I think X-ray crystal structures are the ground truth. There are not very many of them, and they tend to be of the ribosome. If you're looking at secondary structure, covariation is amazing. It's not always the full story, as you saw in some of those secondary structures that had to be improved with 3D. But a really good alignment will tell you a lot about the secondary structure and the base pairs. It's hard to make them, though. It hasn't been automated yet, but it totally can be. It just hasn't been done. So.
Hello.
Hello.
Hi. Nice talk, of course. So I was wondering, like, it's not only the small RNAs, right? It interacts with other RNAs, and then you have RNA binding proteins, other complexes. Like, it's not in simple RNA, right? It uses a lot of a grammar. It creates a paragraph, like, sort of. So what do you think, like, how could you, like, integrate everything and, like, make it approachable?
I don't know about approachable, because I think if you're trying to represent all the complexity of biology, you can't really simplify it at some point. But yeah, I feel like that's a thing where you have to give the user a lot of power to, you have some data model which will be basically uninterpretable to anyone but an expert. You're going to have to give the user an ability to pull out the bits they care about and look at them in reasonable ways. I actually think this is something that LLMs should be doing. So text, in the way we use LLMs, right, is we look at text that it produces. That's awful. Like, it's a great technology because we are all literate. It's thousands of years of practice with it. But I'd rather look at a genome browser when you're telling me what's nearby, right? I'd rather look at a 3D structure when you're talking about contacts. So it would be amazing if there was an LLM that kind of worked across this really rich data domain and then knew to represent this to a user, I should show them, a genome browser, a secondary structure viewer, kind of what it is. But that's very speculative, to put it nicely. Yeah, hopefully that answers your question.
Hi, great talk. Thanks very much for that. I was wondering, when you mentioned the decision making for the LM, you said you were worried about the yes or no because it can hallucinate. And then you asked specifically for it to retrieve a paragraph or whatever from the paper. I was wondering if it can hallucinate there as well. And I think that question came up to me because I was thinking of, again, the decision making from the human side. Said, if the link RNA or the RNA is only predicted computationally, that's immediately removed from any possible annotation. So in that sense, we're not trusting computational predictions. But when you're using, and I understand that these are completely different methods, but when you're using computational methods to annotate, you can trust it. So I started brainstorming that in my head.
Yeah, that's a good point. It is a bit contradictory and possibly, a little too fond of our own work. Right now, there are a lot of Go annotations which are computationally predicted, and right now they're being treated as that. So that's fine, because you'll still have a human who will go eventually and validate it and mark it as, yeah, no, this is totally, this is real, a human did it. As for the idea of hallucinating text, actually, because of constrained decoding, that cannot happen. You can think of it as like a really aggressive brain surgery on the LLM to prevent it from knowing about tokens that you don't want. So it can only produce strings which are found in the original paper. This does drive it a bit crazy. Sometimes it just doesn't work. Andrew has told me cases where it just repeats the word all, because it wants to answer, effectively it wants to say, all of this paper is about that, something like that. But that phrase never appears in the paper, but the word all does. So it just says all. So it has some consequences to LLMs. It's not quite what they're meant to do, but it is very powerful.
I think we have to move on to our short talks.
Thank you all.